huge pleasure for me to be here. Thank you so much to the organizers for giving me the opportunity to come here and talk to you today. The Python community is very near and dear to my heart, and I hope through my talk you can see the application of all the incredible tools that are built. If you have any questions, and apologies in advance for a lot of jargon that's going to come on the slides, take a note, and I'm here for the rest of today and tomorrow, so come and find me. Happy to chat about it. So what I'm going to do today is to try to take you on a journey from building large-scale data sets to power efforts like the Human Cell Atlas, to deploying state-of-the-art AI and machine learning to transform drug discovery, and then we will end on the importance of benchmarking in the open source community. So most of the biomedical research aims to treat or manage disease conditions. And to accomplish that, we need to understand what does it mean to be healthy. Because then when we know that, we have a health reference, we can then infer what is wrong and intervene. So with that in mind, I've led some efforts to build reference data sets for different species that can be used to compare disease atlas or perturbation data sets. And these are built at single-cell transcriptomics resolution. So why do we want to do transcriptomics? First, let me see if I have I don't have a pointer. So transcriptomics, it's the easiest one to measure. The important thing is that unlike all the genome that is the same in every cell of the organism, the transcriptome is incredibly variable cell to cell. And so it offers a close representation of cell states within the organs. And then why do we want to do it at single cell? Like the picture in here is trying to convey, if we would only look in the bulk or if we would only look in the juice, it's very hard to tell if a bad fruit has made it in, or like which of the fruits was bad. And so if we look at each of them individually, then we have actually the opportunity of trying to understand which cell populations might be the ones who are causing the disease. And so if you're wondering what single-cell transcriptomics data looks like, it's for the most part a matrix that is 50,000 rows that are the genes and as many cells that can go in the order of, like these days we're working with 100 to 200 million individual cell transcriptomics. And so these organism cell atlas are really a key piece to understanding the underlying mechanisms of the disease because they serve the reference point to the non-disease state. There is a commonality across all the projects that I'm going to show you today, which is how we process the data. So we started with the mouse, which was tabula muris, which is the reference state. And then we explored the mechanisms of natural aging when we built tabula muricinis, which expanded the mapping of all the cells in the mouse from the 3 months to from 1 month to 30 months. And then we looked at what would look like an intervention. So parabiosis is this experimental system where you connect the blood circulation of two animals, the old animal and the young animal, and you look for rejuvenation effect. So it's an intervention that you can do. And we took this paradigm of looking through different stages from references to diseases and interventions across different species. And so this is the catalog of the data that is all available. I'll show you the details. Tabula sapiens, which is one of the first references for the Human Cell Atlas, but the way that all the data has been shared in code applies across all these projects. So in Tabula sapiens, we try to get as many tissues as possible from a single donor. So we have a network of experts that dissociates each of the tissues, and then we partner with Donor Network West to help us procure the donors with all the ethical consent. From there, we go into cell dissociation. So for each of the organs in the body, the liver, the heart, the muscle, we have a team that has expertise in that particular tissue. And so after that, it gets all centralized. So decentralization effort is important because it reduces the technical artifacts. Whenever we are doing experiments, there is always confounding effects when the experiments are done. It's technical variability. We have to try to minimize it, to work with it, to correct. But if we can, a priori, when we are designing the experiments, preemptively address it, it's usually a positive thing to minimize and to have better data quality downstream. So we centralize all these pieces and we centralize all the data analysis. And so here is when we start getting into code. And I'm not going to show any scripts today, but just going to walk you through the different steps. So we have our matrices, so kind of like remember that matrices that is millions of cells for about 50,000 rows of genes. We process this data to remove ambient RNA. So when we are dissociating the cells, so we're isolating cells, there is always some of those cells we're going to burst. And they release the material they have inside, which is an RNA that we are trying to collect. But the problem is, remember, we're doing at single cell resolution, so we do not want the RNA of those cells that burst because it doesn't belong to the cells that we are profiling. So computationally, we can develop algorithms to remove the interference of the RNA of the other cells. And then we try to integrate everything so that we bring it into a single latent space. And so at the first pass, what we get is this single representation of the whole body. So these are all the cells. Each dot in this plot is one cell. It's color-coded by the tissue of origin because these are labels that we know at the beginning. But then we can visualize other labels that came with the data that we know, like what is the donor, what is the compartment, is it male or female. But there is a lot of things that we still don't know at this point. We can kind of like try to look at the overall atlas and see for across the donors, from which donors were we able to manage more tissues, what is like the complete representation. And then one thing that we were really interested, because it's very important for drug discovery, is to also have an idea of clinical metadata. This means the variables that are part of the life of these donors, their age, their gender. So I was walking you through kind of like just the overview of the summary of the data set. And one thing that I want to call out here is this is a growing data set because we can continue accumulating data, and I'll share a bit more in the next slides. But so for V1, where we were with about 500,000 cells and sharing the clinical metadata side by side with the data. But one of the things that we are really trying to understand is this single cell resolution, and for that, we do need cell types. So when we look at this blob, we do not know if the cells in the blob are an hepatocyte, a cell of your liver, are a cell of the immune system, are a cell of the muscle. So we really need to embark on this journey of what is called cell type annotation. And this is one of the most challenging tasks in single cell data analysis. So what we have done in Tabula Sapiens is to try to take a strategic approach where we leverage previously annotated data to accelerate the annotations on this incredibly time-consuming step. And so how does that look like? So on the data summary that I showed you, we had 15 donors. So when we got donor 1, we did manual cell type annotation. This is looking through all these data matrix and identifying genes that are specific to clusters. And so we identify that, we build the reference, and then comes donor 2, which is now a completely unclassified data set. And so what we want to do is we want to find a way of using this draft reference to classify the new data, and then we engage with the experts in curation such that we have an updated reference that is curated. We are building a data set for the community, so we want to make sure that the labels that are provided are of high quality. And so we did this through few loops, and then we noticed that a lot of methods in the community, so a lot of publications are about, oh, this is my new method, this method is better than these previous methods. So we thought, okay, so why don't we take all those methods and create a statistical framework that we call popular vote, PopV for short, that what basically PopV does under the hood is it gets the input of the annotated query data set, it has a reference, and then it runs 8 methods, but it is scalable, so all the code more methods can be added, and then we output reports with statistics on the agreement between the methods and other key metrics. So now we have a method that can take advantage of all these best-in-class, new best method that keeps being published. And so we can go back in here and quickly try to move into a new reference, which is what was in the publication. And so now that we have these cell types that have been annotated, we can go back to our atlas and start digging through cell types. So let's look through the macrophages as an example. So macrophages are cells of the immune system that are involved in fighting infection. And they are typically found in many tissues, so this is very rewarding to see that on the embedding space, they are co-clustering, so that means they are sharing some properties. Then we see them colored by different tissues, meaning that we were able to find them across the body. And so we can look at all these cells in here. We can look at correlations across the organs, so we can see that some macrophages are more similar within some groups of tissues. We can look if there are genes, so these are like on the left of the violin heat map. You have the names of genes. Some genes are only expressed in some tissues, others are not. And so it really starts to give us an idea of specificity of gene programs and cell types, and this has implications then on how we are going to think about therapies, for example. And so when we have all this data, we can do many more investigations. The publications have more. I'm not going to go in more details here for the benefit of time. But this is only one layer of information, because we've basically taken a tissue, we've, for the most part, mashed it, imagining kind of like putting it through a blender, and then you are collecting transcriptomics at the end. But we all know that we have spatial organization. So what we've also collected is all the images of histology, and we have also tissue that could be used for spatial profiling, and also another important component of the human cellular fraction, which is the microbiome, because we have indeed a lot of microbiome cells. And so this was what was in the first version of the references, which was published back in 2022. And since then, we have continued to accumulate data. So in V2, which we posted on bioRxiv and made all the data available at the end of last year, we now have grown into about 1.2 million cells. We have increased the number of tissues, the number of donors, and the number of cell types. And so this continues to grow as one of the contributing projects for the Human Cell Atlas, which is a global effort across the world to really map all the cells. So it's, for those of you who might be familiar with the human genome, the Human Cell Atlas is kind of like the next step, building on top of the human genome. Now that we know what are the genes across all the cells, how do these genes get expressed? How do they function? So this is the next level of iteration. And when we were building V2, we had the luxury on top of the data that we had collected to get access to the electronic medical records. So they have been fully de-anonymized. So this offers another layer of information that can be really helpful for building more methods and for like data science and machine learning. And if you would want to interact with the data, you can go to an application, you can get the link from the data portal where you can chat with the Tabula Sapiens electronic medical records. And all this data is available, is easily reached from our data portal where you have kind of like links into everything. I've put a lot of effort in democratizing the access to these large-scale resources, and I really think they are the equivalent of a gold mine for biomedical research. And then if you are more interested of like, can I just kind of like get a data matrix, start working, if you go to CZI cell by gene or through the Census API, all the data is ready to download, and this is immediately compatible with the typical single-cell analysis packages in Python and R. And so I strongly believe that the impact of this work depends on making it accessible to the community. So in summary, there's all these ways that you can get your hands on the data, and although I focus my talk on Tabula sapiens, there's a very similar way of getting access to all the other data sets. So if you have any questions, you can find it, feel free to ping me, and I'll send you through the links. It takes a large consortium. There's over 160 people building Tabula sapiens, and it could not have happened with the support of the John Zuckerberg BioHub, Stanford, Berkeley, UCSF, CZI, and the donor network West. And so in the first part of my talk, I've walked you through steps of building a data resource, which supports our quest in understanding disease. And what AI can really do is, or what we can do with AI, is to leverage all these types of data and help us advance drug discovery. So, I mean, we've all seen the complex tasks that AI is able to do, but I think the question continues to be, can it really help us in the important task of making better medicines? And we are at an earlier stage of this exponential curve, but I really think that with this right data and the right people building models, we can move ourselves forward. So what I'm going to do now is to walk you through a hands-on application of deploying AI/ML in the context of drug discovery. So at insitro, which is the company that I am now, we leverage the increasing wealth of rich clinical data modalities that are routinely collected and enable us to define precise phenotypic traits, to get novel therapeutic insights. We integrate these with human genetics, because this has been shown that drugs that have human genetic support have a higher probability of succeeding in making it into the clinic. And then we build conviction around our targets by doing specific knockouts. So when you knock out a gene is when you're basically removing that gene from those lines and then understanding what are the consequences of that gene not being present. And we collect a range of measurements across different modalities that really help us understand what does the phenotype look like. And so one of the diseases that we've been focusing on is ALS. It's a collaboration with our partners at Bristol Myers Squibb, who have been critical for the support of this project. And we have established the largest bank of iPSC, that is induced pluripotent stem cells, derived motor neurons from patients with ALS. And we are using it to try to discover new targets. So how do we go about it? We have cellular models, so these are cell lines. We got cells from patients with ALS in the lab. We can reprogram them to become a stem cell, so a cell that we can differentiate into cells that have the properties that we wanted. For ALS is the motor neurons, which are the neurons that control movement. And so we have representation of 8 mutations that are known to cause familial ALS. And then we leverage StatMin2, so try to memorize just this one gene, StatMin2, which is a gene that basically is used as a biomarker because there's a protein which is a hallmark of ALS. It's called TDP43. When there is disease, that protein moves from the nucleus to the cytoplasm. When that protein is in the cytoplasm, you have different RNA species for StatMin2. And so this is why we can use it as a biomarker. And so a healthy cell is going to have nucleotide TDP43, very good StatMin2, 6-cell cytoplasmatic TDP43 cryptic exons on StatMin2. And this is what is known to be really associated with the disease. And so after that, we leverage self-supervised machine learning models that create embeddings to capture relevant biological information. Without going into too much detail, for those in the audience who might not be super familiar with the self-supervised learning, it's an ML training paradigm that we provide data without labels. And then the model uses subsets and augmentations of the input data to learn which inputs belong to the same class. And we have been leveraging extensively the Dino VIT architecture, which combines the concepts that are on this slide to help us classify images on this a large unlabeled data set. One of the things that we have noticed when we deploy these models, you can get these models trained from the internet, but they are typically trained in data sets that are not sensitive for biological applications. They might be trained on ImageNet, which is kind of like the collection of digits, but when we are looking into cell biology, the subtleties of the morphology of those cells is very much what we are after. And so we ended up having to train our models in-house from scratch, and you can see that the accuracy is really improving with our train on the plot in the right. And so then, after we have our cell models, we have the machine learning stack that we want to work with. We designed 3 tasks to test if the models and the corresponding embeddings are actually capturing ALS-relevant biological information. So we trained models to predict the disease state. So that's the first task, predict healthy from sick or wild type from mutant. Predict the classical features. So this is the TDP43 localization that I was telling you about, when it's like either in the nucleus or on the cytoplasm. And then the hardest task that we tried to do is to go multimodal. So we have measurements of transcriptomics, we have measurements on imaging. We try to predict the transcriptomics, so the gene expression, from the morphology. In the first biological credentialing task to predict disease state, we define that by the ability to classify, so we build a classifier from mutant to wild type. And for each genetic mutation, we held out one donor and we cross-validate. And then we make sure each class and the donor background is balanced. And then we do the same thing across the mutations, because we have the cell models for different mutations. And so in comparison with the classical measurement of TDP43 mislocalization, which is in black or grayish, and the ML classifier in blue, we observe that the ML embeddings are really increasing the accuracy in distinguishing the mutant from the wild type. And then we took the next step and decided to impute the relative abundance of TDP43. So from the model, so this is like on the images that we have trained, we have measured a lot of this protein, we are now trying to impute the relative abundance of TDP43. And we found really good correlation with the measured ratios. So this is kind of like what we have here on the right, and this was like the second task that we had set to do, which was to credential the model to predict these classical features. So together, we found very compelling evidence that our embeddings were capturing disease-relevant biology. So we took the next step into the last task, which was to impute transcriptomics from image embeddings. And so the question here is really whether the embeddings that were trained only on imaging were able to predict gene expression. So when we construct the latent spaces using Dino, no transcriptomic information is shared with the model. It's trained only on the images. We then have companion gene expression, so we can train a model that tries to learn from these embeddings of the images into the matrices of gene expression. So we selected imaging and RNA-seq data from 40 unique conditions for treatment, and we wanted to have like sufficient quality for paired analysis. We collected the RNA at the well level, so this was not done single cell, unfortunately, so we only have a well measurement. This is on a cell line. Cell lines tend to be more homogeneous. Imaging, though, is collected at the single cell because we can segment, right? So we can have all the information, but we had to aggregate the embeddings. So we calculated mean morphological embedding for all the image tiles. We had the gene expression for all the RNA-seq replicates. And then we reduce the components that were explainable by cell count with an applied dimensionality reduction techniques, extracting the top 50 principal components. And then finally, we trained a multitask lasso regression model that predicted the expression residual of each gene from the projection of each embedding vector. And so this is the setup that we had. And so, okay, so when we looked at the predictability of ALS genes, so are the embeddings able to predict all the genes? This is what we have in blue. Are the embeddings able to predict genes that have been known to be associated with the disease ALS? We found that the predictability of the genes implicated in ALS was higher, yet again suggesting that the embeddings are carrying some biological information. And so then what we tried to do was, okay, so now that we have this ability of predicting gene expression, can we impute the StatMin2 expression in disease lines and compared with what had been measured in the experiments? And so this is what you have here in this graph. So the dino imputed is the first row. This is across two donors. In blue is the disease line. In orange is the corrected line. So it's the equivalent of a healthy. And you can see that the model is able to recapitulate the trend. The magnitude is not the same, but the trend is in there, which for us was very reassuring because we were really trying to figure out how much value could we extract from these embeddings. And then finally, what we did was to impute differential gene expression between the disease and the wild type lines and compare this imputed differential gene expression with what we have measured. We also found good correlation. So with all these credential tasks done for the models, we felt that we were then ready to start going forward in identifying novel targets in ALS. So we have these over 200 isogenic APSC lines. We have a way of generating the motor neurons from the donor lines, and we can capture and analyze these multimodal phenotypic data at scale. So we are ready to screen. But what does it mean to screen? So when we are thinking about building a screen, what we are in general imagining is if I perturb, either I do a drug treatment or I do an environmental stress or I do a genetic treatment, what are the consequences? And can I pinpoint it to one gene, one molecule that is going to be my next target? And for us, screening at this stage for target discovery very commonly means genetic screening. So genetic screening is when we are going to introduce a knockout. We're going to remove a gene, and then we are going to measure the behavior of that cell after we have removed the gene, and then identify if the behavior of, let's say we do the knockout in a disease line, and then so that gene is no longer there, and then we are going to measure the behavior of the cell. The question that we ask is the behavior of this cell that I just removed the gene now closer to my healthy cell? So is my disease cell starting to behave more like a healthy cell. And this is how you screen in vitro to identify novel drug targets. And in vitro, we do this in a pooled manner. So that means that instead of doing one knockout in each well, we have a bulk, like we have a pool of knockouts. We have a library that gets used to infect cells. So this is like done in kind of like a large vessel. And so we try to do this at a point that we optimize for one cell only getting one genetic edit. And then we do optical phenotyping. So these edits are barcoded. So then we can put the cells under the microscope and we can image to identify the barcodes. And this is how we know which perturbation the cells have. If they happen to have 2 perturbations, we can remove them when we are processing the data, because we will know that for that cell we have 2 barcodes, so that is not data that at the moment we are interpreting. There are works in the field that are starting to look at the effects of combinatorics, so when you do like multiple perturbations in one cell, but at the moment we are focusing on like we only want to analyze cells that have a single perturbation, so that's the part of kind of like the sequencing. So we then build these phenotype to genotype matrix, and we look for our drivers and our modifiers. And this technique has been published before by a lot of groups. There's especially active area of research at the Broad Institute. What we have done at insitro is the productionization. So we want to really be able to run it at scale. So we have invested on the hardware, the software, and the machine learning. You can see, hopefully the video is playing, yes. You can see like the robotic arm in there moving the plates around, because to scale something like this is a massive investment in microscopy, automation, chemistry, assay design, data pipelines, machine learning. It's basically, we need to do multiple steps of the in situ sequencing. And so having this automated is really critical to have high quality data generation. It takes a village. It takes a whole startup to build something like that at the beginning, and then you can scale. And so we have the system ready to be deployed. If we go back to our kind of like original diagram of how we've laid out from kind of like modeling, the phenotyping, the discovery, the ML. So now what we are going to do is to do this pooled screen. So we have kind of like a library of genes. We're going to screen. We're going to find genes. And so basically, then what we have in here, I cannot disclose the names of the targets, but what we have in here is then one of the results of looking through this screen and identifying new targets. And there has been a press release that you can check on the QR code about the new target that we have identified for ALS, that we are now going into experimental validation and hopefully into drug discovery. And we're really very excited that we've been able to deploy machine learning all the way to identify new targets on this devastating disease. Majority of the work that I've highlighted is contained on these 2 preprints. If you want to take a look, and if you want to learn more about in situ, the work we do, careers, internships, check out our website. And so I hope I have convinced you that high content data are already being used in drug discovery. The reference atlas that I started at the beginning is, among other things, supporting the development of appropriate cellular models, because this is how we know what the cell that we are building in the lab should look like when we have the reference cells that we take from the donors. And then what we have done at insitro is based on high content cellular data from the design for purpose cell models. And we found that machine learning is able to identify subtle patterns across different assay modalities that provide a more holistic view of the underlying biology. And so now that I've walked you through the different steps of the analysis, I know very much kind of like no code has been mentioned. I want to call it that it's important to notice that we continuously need newer and better tools. And this is where I'm inviting you all to contribute. So the number of tools that has been published that's used to perform analysis is for the most part getting kind of like close to an exponential growth. So back in 2020, myself with a group of colleagues from around the world, we also work in the space of single-cell transcriptomics, data science, and machine learning. We founded open problems in single-cell analysis. And what we are trying to address in here is the problem of decentralized benchmarking. Very much like I was mentioning what motivated us to build Popular Vote, PopV for cell type annotation. Every method developer is going to compare their methods to others that are out there. So they're going to do this independently. And then they are going to optimize the parameters of the baseline models. They're going to do some improvements on the pre-processing. And so according to the papers, the methods are always better. But when you look at it, in reality, maybe not so much. And there is also another challenge, which is even publications like the ones that are highlighted in here, that are publications that specifically try to benchmark methods. They are going to age very quickly because the publication is a snapshot in time. And so very often, after the authors have published, which is what is in green, they are no longer updating the code, or they might even do one code update. But after that, there is no incentive for the authors of those publications to have this as a living resource. So what we try to do with open problems is to provide these centralized benchmarking data sets and community events that fuel innovation. So there's a very active interdisciplinary community. There's a Discord server. We host community meetings, weekly co-working meetings. We continuously update, and it is centralized. So we have a platform that the developers and the users can contribute data sets, methods, metrics. Building this benchmarking platform requires a very diverse tech stack, includes workflow orchestration, cloud computing, unit testing frameworks, schema, API, definitions. And so all of these have a steep learning curve. And what we try to do with the platform is to build a system that is tolerant to users that do not have expert knowledge on every component of the stack. So we really want to make it easy for people to contribute. And then we have done innovative data sets and international competition. So we've generated data to kind of like really power this competition. So we have run competitions at NeurIPS. I think we were the first ones to ever run like a single cell transcriptomics competition at NeurIPS because the ML community hasn't really been looking into single cell data. We did a data generation for that. We did another multimodal time course, and then we did a perturbational data set. This data is available from our website. And we're very proud to see all the publications that are not by us, so these we are not authors on these papers, that have kind of like been enabled by the platform. At the moment, we have benchmarks for 12 tasks, and we're counting. If you have an idea, if you want to contribute a task, please do reach out. We really want this to be a living benchmarking, a living platform. I'm part of the core team. I'm by no means the main driver. All these fantastic people on the box in there have done majority of the work. We have the task contributors. We have scientific advisors and sponsors. And it just so happened that the milestone that we were very much waiting for, which was like to have a publication that people can cite, like a publication that is about having a platform, came out on Tuesday. And we were like, okay, I'm going to kind of like make sure I give just kind of like a shout out to all the amount of work that got done in there. And so we would love to have you join us, get involved. It should be very straightforward. We would love to kind of like have more contributions, have engagement. And with this, I hope I have convinced you that data-driven scientific research is collaborative, is computational, and we are really starting to reshape on how we think about understanding the mechanisms of disease and doing drug discovery. And thank you so much. Thank you so much for having me. And if you have any questions, be happy to take them.